16.4 Eukaryotic transcription gene regulation. By the end of this section, you will be able to do the following: discuss the role of transcription factors in gene regulation, explain how enhancers and repressors regulate gene expression like prokaryotic cells. The transcription of genes in eukaryotes requires the action of an RNA polymerase to bind to a DNA sequence upstream of a gene in order to initiate transcription. However, unlike prokaryotic cells, the eukaryotic RNA polymerase requires other proteins, or transcription factors, to facilitate transcription initiation. RNA polymerase by itself cannot initiate transcription in eukaryotic cells. There are two types of transcription factors that regulate eukaryotic transcription. General, or basal, transcription factors bind to the core promoter region to assist with the binding of RNA polymerase. Specific transcription factors bind to various regions outside of the core promoter region and interact with the proteins at the core promoter to enhance or repress the activity of the polymerase. Link to learning. View the process of transcription, the making of RNA from a DNA template. Click to view content, https://www.openstax.org/l/transcript_RNA. The promoter and the transcription machinery genes are organized to make the control of gene expression easier. The promoter region is immediately upstream of the coding sequence. This region can be short, only a few nucleotides in length, or quite long, hundreds of nucleotides long. The longer the promoter, the more available space for proteins to bind. This also adds more control to the transcription process. The length of the promoter is gene-specific and can differ dramatically between genes. Consequently, the level of control of gene expression can also differ quite dramatically between genes. The purpose of the promoter is to bind transcription factors that control the initiation of transcription. Within the core promoter region, 25 to 35 bases upstream of the transcriptional start site, resides the TATA box. The TATA box has the consensus sequence of 5' TATAAA3. The TATA box is the binding site for a protein complex called TFIID, which contains a TATA binding protein. Binding of TFIID recruits other transcription factors, including TFIIB, TFIIE, TFIIF, and TFIIH. Some of these transcription factors help to bind the RNA polymerase to the promoter, and others help to activate the transcription initiation complex. In addition to the TATA box, other binding sites are found in some promoters. Some biologists prefer to restrict the range of the eukaryotic promoter to the core promoter, or polymerase binding site, and refer to these additional sites as promoter proximal elements, because they are usually found within a few hundred base pairs upstream of the transcriptional start site. Examples of these elements are the CAAT box, with the consensus sequence 5' CCAAT3, and the GC box, with the consensus sequence 5' GGGCGG3. Specific transcription factors can bind to these promoter proximal elements to regulate gene transcription. A given gene may have its own combination of these specific transcription factor binding sites. There are hundreds of transcription factors in a cell, each of which binds specifically to a particular DNA sequence motif. When transcription factors bind to the promoter just upstream of the encoded gene, it is referred to as a cis-acting element, because it is on the same chromosome just next to the gene. Transcription factors respond to environmental stimuli that cause the proteins to find their binding sites and initiate transcription of the gene that is needed. Enhancers and transcription. In some eukaryotic genes, there are additional regions that help increase or enhance transcription. These regions, called enhancers, are not necessarily close to the genes they enhance. They can be located upstream of a gene, within the coding region of the gene, downstream of a gene, or maybe thousands of nucleotides away. Enhancer regions are binding sequences, or sites, for specific transcription factors. When a protein transcription factor binds to 404 Chapter 16, Gene Expression Access for free at OpenStax.org. Its enhancer sequence, the shape of the protein changes, allowing it to interact with proteins at the promoter site. However, since the enhancer region may be distant from the promoter, the DNA must bend to allow the proteins at the two sites to come into contact. DNA bending proteins help to bend the DNA and bring the enhancer and promoter regions together. Figure 16.9. This shape change allows for the interaction of the specific activator proteins bound to the enhancers with the general transcription factors bound to the promoter region and the RNA polymerase. Figure 16.9 Interaction between proteins at the promoter and enhancer sites. An enhancer is a DNA sequence that promotes transcription. Each enhancer is made up of short DNA sequences called distal control elements. Activators bound to the distal control elements interact with mediator proteins and transcription factors. Two different genes may have the same promoter but different distal control elements, enabling differential gene expression. Turning genes off. Transcriptional repressors like prokaryotic cells. Eukaryotic cells also have mechanisms to prevent transcription. Transcriptional repressors can bind to promoter or enhancer regions and block transcription. Like the transcriptional activators, repressors respond to external stimuli to prevent the binding of activating transcription factors. 16.5 Eukaryotic post-transcriptional gene regulation. By the end of this section, you will be able to do the following. Understand RNA splicing and explain its role in regulating gene expression. Describe the importance of RNA stability in gene regulation. RNA is transcribed, but must be processed into a mature form before translation can begin. This processing that takes place after an RNA molecule has been transcribed, but before it is translated into a protein, is called post-transcriptional modification. As with the epigenetic and transcriptional stages of processing, this post-transcriptional step can also be regulated to control gene expression in the cell. If the RNA is not processed, shuttled, or translated, then no protein will be synthesized. RNA splicing, the first stage of post-transcriptional control in eukaryotic cells. The RNA transcript often contains regions, called introns, that are removed prior to translation. The regions of RNA that code for protein are called exons. Figure 16.10. After an RNA molecule has been transcribed, but prior to its departure from the nucleus to be translated, the RNA is processed and the introns are removed by splicing. Splicing is done by 16.5, eukaryotic post-transcriptional gene regulation. 405 spliceosomes, ribonucleoprotein complexes that can recognize the two ends of the intron, cut the transcript at those 2 points, and bring the exons together for ligation. Figure 16.10 Pre-mRNA can be alternatively spliced to create different proteins. Evolution connection alternative RNA splicing. In the 1970s, genes were first observed that exhibited alternative RNA splicing. Alternative RNA splicing is a mechanism that allows different protein products to be produced from one gene when different combinations of exons are combined to form the mRNA. Figure 16.11. This alternative splicing can be haphazard, but more often it is controlled and acts as a mechanism of gene regulation, with the frequency of different splicing alternatives controlled by the cell as a way to control the production of different protein products in different cells or at different stages of development. Alternative splicing is now understood to be a common mechanism of gene regulation in eukaryotes. According to one estimate, 70% of genes in humans are expressed as multiple proteins through alternative splicing. Although there are multiple ways to alternatively splice RNA transcripts, the original 5'-3" order of the exons is always conserved. That is, a transcript with exons 1, 2, 3, 4, 5, 6, 7 might be spliced 1, 2, 4, 5, 6, 7 or 1, 2, 3, 6, 7, but never 1, 2, 5, 4, 3, 6, 7. Figure 16.11. There are 5 basic modes of alternative splicing. How could alternative splicing evolve? Introns have a beginning and ending recognition sequence. It is easy to imagine the failure of the splicing mechanism to identify the end of an intron and instead find the end of the next intron, thus removing 2 introns and the intervening exon. In fact, there are mechanisms in place to prevent such intron skipping, but mutations are likely to lead to their failure. Such mistakes would more than likely produce a non-functional protein. Indeed, the cause of 406 Chapter 16, Gene Expression Access for free at OpenStax.org. Many genetic diseases as abnormal splicing rather than mutations in a coding sequence. However, alternative splicing could possibly create a protein variant without the loss of the original protein, opening up possibilities for adaptation of the new variant to new functions. Gene duplication has played an important role in the evolution of new functions in a similar way by providing genes that may evolve without eliminating the original, functional protein. Question. In the corn snake Pantherophis gutatus, there are several different color variants, including amelanistic snakes whose skin patterns display only red and yellow pigments. The cause of amelanism in these snakes was recently identified as the insertion of a transposable element into an intron in the OCA2, oculocutaneous albinism, gene. How might the insertion of extra genetic material into an intron lead to a non-functional protein? Link to learning. Visualize how mRNA splicing happens by watching the process in action in this video. Click to view content, https://www.openstax.org/l/mRNA_splicing control of RNA stability before the mRNA leaves the nucleus. It is given two protective caps that prevent the ends of the strand from degrading during its journey. 5' and 3' exonucleases can degrade unprotected RNAs. The 5' cap, which is placed on the 5' end of the mRNA, is usually composed of a methylated guanosine triphosphate molecule, GTP. The GTP is placed backward on the 5' end of the mRNA, so that the 5' carbons of the GTP and the terminal nucleotide are linked through 3 phosphates. The poly A tail, which is attached to the 3' end, is usually composed of a long chain of adenine nucleotides. These changes protect the two ends of the RNA from exonuclease attack. Once the RNA is transported to the cytoplasm, the length of time that the RNA resides there can be controlled. Each RNA molecule has a defined lifespan and decays at a specific rate. This rate of decay can influence how much protein is in the cell. If the decay rate is increased, the RNA will not exist in the cytoplasm as long, shortening the time available for translation of the mRNA to occur. Conversely, if the rate of decay is decreased, the mRNA molecule will reside in the cytoplasm longer and more protein can be translated. This rate of decay is referred to as the RNA stability. If the RNA is stable, it will be detected for longer periods of time in the cytoplasm. Binding of proteins to the RNA can also influence its stability. Proteins called RNA-binding proteins, or RBPs, can bind to the regions of the mRNA just upstream or downstream of the protein coding region. These regions in the RNA that are not translated into protein are called the untranslated regions, or UTRs. They are not introns, those have been removed in the nucleus. Rather, these are regions that regulate mRNA localization, stability, and protein translation. The region just before the protein coding region is called the 5' UTR, whereas the region after the coding region is called the 3' UTR, figure 16.12. The binding of RBPs to these regions can increase or decrease the stability of an RNA molecule, depending on the specific RBP that binds. Figure 16.12 RNA binding proteins. The protein coding region of this processed mRNA is flanked by 5' and 3' untranslated regions, UTRs. The presence of RNA binding proteins at the 5' or 3' UTR influences the stability of the RNA molecule. RNA stability and microRNAs, in addition to RBPs that bind to and control, increase or decrease RNA stability. Other elements called microRNAs can bind to the RNA molecule. These microRNAs, or mRNAs, are short RNA molecules that are only 21 to 24 nucleotides in length. The mRNAs are made in the nucleus as longer pre-mRNAs. These pre-mRNAs are chopped into mature mRNAs by a protein called dicer. Like transcription factors and RBPs, mature mRNAs recognize a specific sequence and bind to the RNA. However, mRNAs also associate with a ribonucleoprotein complex called the RNA-induced silencing complex, RISC. The RNA component of the RISC base pairs with complementary sequences on an mRNA and either impede translation of the message 16.5, eukaryotic post-transcriptional gene regulation 407, or lead to the degradation of the mRNA.